Collagen is the main structural protein of the various connective tissues in animals. As the main component of connective tissue, it is the most abundant protein in mammals, making up from 25% to 35% of the whole body protein content. Collagen, in the form of elongated fibrils, is mostly found in fibrous tissues such as tendons, ligaments and skin. It is also abundant in corneas, cartilage, bones, blood vessels, the gut, intervertebral discs and the dentin in teeth. In muscle tissue, it serves as a major component of the endomysium. Collagen constitutes 1 to 2% of muscle tissue, and accounts for 6% of the weight of strong, tendinous muscles. The fibroblast is the most common cell that creates collagen. Gelatin, which is used in food and industry, is collagen that has been irreversibly hydrolyzed. Collagen also has many medical uses in treating complications of the bones and skin. The name collagen comes from the Greek kolla meaning glue, and suffix gen denoting producing. This refers to the compound's early use in the process of boiling the skin and sinews of horses and other animals to obtain glue. History and background. The molecular and packing structures of collagen have eluded scientists over decades of research. The first evidence that it possesses a regular structure at the molecular level was presented in the mid-1930s. Since that time, many prominent scholars, including Nobel laureates Crick, Pauling, Richendranath, and others, including Brodsky, Berman, and Ramachandran, concentrated on the conformation of the collagen monomer. Several competing models, although correctly dealing with the conformation of each individual peptide chain, gave way to the triple helical Madras model of Ramachandran, which provided an essentially correct model of the molecule's quaternary structure, although this model still required some refinement. Not clear which refinement. Question: the packing structure of collagen has not been defined to the same degree outside of the fibrillar collagen types, although it has been long known to be hexagonal or quasi-hexagonal. As with its monomeric structure, several conflicting models alleged that either the packing arrangement of collagen molecules is sheet-like or microfibrillar. The microfibrillar structure of collagen fibrils in tendon, cornea and cartilage has been directly imaged by electron microscopy. The microfibrillar structure of tail tendon, as described by Fraser, Miller, and Wess, was modeled as being closest to the observed structure, although it oversimplified the topological progression of neighboring collagen molecules, and hence did not predict the correct conformation of the discontinuous d-periodic pentameric arrangement termed simply, the microfibril. Various cross-linking agents like dopaquinone, embolin, potassium embolate and 5-O-methyl-embolin could be developed as potential cross-linking stabilization agents of collagen preparation, and its application as wound dressing sheet in clinical applications is enhanced. Chemistry. Collagen is composed of a triple helix, which generally consists of two identical chains and an additional chain that differs slightly in its chemical composition. The amino acid composition of collagen is atypical for proteins, particularly with respect to its high hydroxyproline content. The most common motifs in the amino acid sequence of collagen are glycine proline X and glycine X hydroxyproline, where X is any amino acid other than glycine, proline or hydroxyproline. The average amino acid composition for fish and mammal skin is given. Synthesis. First, a three-dimensional stranded structure is assembled, with the amino acids glycine and proline as its principal components. This is not yet collagen but its precursor, precollagen. A recent study, Murad et al., 1981, shows that vitamin C must have an important role in its synthesis. Prolonged exposure of cultures of human connective tissue cells to ascorbate induced an 8-fold increase in the synthesis of collagen with no increase in the rate of synthesis of other proteins. Since the production of precollagen must precede the production of collagen, vitamin C must have a role in this step. The conversion involves a reaction that substitutes a hydroxyl group, OH, for a hydrogen atom, H, in the proline residues at certain points in the polypeptide chains, converting those residues to hydroxyproline. This hydroxylation reaction organizes the chains in the conformation necessary for them to form a triple helix. The hydroxylation, next, of the residues of the amino acid lysine, transforming them to hydroxylysine, is then needed to permit the cross-linking of the triple helices into the fibers and networks of the tissues. These hydroxylation reactions are catalyzed by two different enzymes, prolyl-4-hydroxylase and lysyl-hydroxylase. Vitamin C also serves with them in inducing these reactions. In this service, one molecule of vitamin C is destroyed for each H replaced by OH. The synthesis of collagen occurs inside and outside of the cell. The formation of collagen which results in fibrillary collagen is discussed here. Meshwork collagen, which is often involved in the formation of filtration systems, is the other form of collagen. All types of collagens are triple helices, and the differences lie in the makeup of the alpha peptides created in step 2. Transcription of mRNA. About 34 genes are associated with collagen formation, each coding for a specific mRNA sequence, and typically have the COL prefix. The beginning of collagen synthesis begins with turning on genes which are associated with the formation of a particular alpha peptide. Pre-pro-peptide formation. Once the final mRNA exits from the cell nucleus and enters into the cytoplasm, it links with the ribosomal subunits and the process of translation occurs. The early first part of the new peptide is known as the signal sequence. The signal sequence on the N-terminal of the peptide is recognized by a signal recognition particle on the endoplasmic reticulum, which will be responsible for directing the pre-pro-peptide into the endoplasmic reticulum. Therefore, once the synthesis of new peptide is finished, it goes directly into the endoplasmic reticulum for post-translational processing. It is now known as pre-pro-collagen. Alpha peptide to procollagen. 3 modifications of the pre-pro-peptide occur leading to the formation of the alpha peptide. Secondly, the triple helix known as procollagen is formed before being transported in a transport vesicle to the Golgi apparatus. 1. The signal peptide on the N-terminal is dissolved, and the molecule is now known as propeptide. 2. Hydroxylation of lysines and prolines on propeptide by the enzymes prolyl hydroxylase and lysyl hydroxylase occurs to aid cross-linking of the alpha peptides. This enzymatic step requires vitamin C as a cofactor. In scurvy, the lack of hydroxylation of prolines and lysines causes a looser triple helix. 3. Glycosylation occurs by adding either glucose or galactose monomers onto the hydroxy groups that were placed onto lysines, but not on prolines. From here the hydroxylated and glycosylated propeptide twists towards the left very tightly and then 3 propeptides will form a triple helix. It is important to remember that this molecule, now known as procollagen, is composed of a twisted portion and 2 loose ends on either end. At this point, the procollagen is packaged into a transfer vesicle destined for the Golgi apparatus. Golgi apparatus modification. In the Golgi apparatus, the procollagen goes through one last post-translational modification before being secreted out of the cell. In this step, oligosaccharides are added, and then the procollagen is packaged into a secretory vesicle destined for the extracellular space. Formation of tropocollagen. Once outside the cell, membrane-bound enzymes known as collagen peptidases remove the loose ends of the procollagen molecule. What is left is known as tropocollagen. Defects in this step produce one of the many collagenopathies known as Ehlers-Danlos syndrome. This step is absent when synthesizing type 3, a type of fibrillar collagen. Formation of the collagen fibril, lysyl oxidase, an extracellular enzyme, produces the final step in the collagen synthesis pathway. This enzyme acts on lysines and hydroxylysines producing aldehyde groups, which will eventually undergo covalent bonding between tropocollagen molecules. This polymer of tropocollagen is known as a collagen fibril. Amino acids. Collagen has an unusual amino acid composition and sequence. Glycine is found at almost every third residue. Proline makes up about 17% of collagen. Collagen contains 2 uncommon derivative amino acids not directly inserted during translation. These amino acids are found at specific locations relative to glycine and are modified post-translationally by different enzymes, both of which require vitamin C as a cofactor. Hydroxyproline derived from proline, hydroxylysine derived from lysine, Depending on the type of collagen, varying numbers of hydroxylysines are glycosylated. Cortisol stimulates degradation of collagen into amino acids. Collagen I formation. Most collagen forms in a similar manner, but the following process is typical for type I. Inside the cell, two types of alpha helices are formed during translation on ribosomes along the rough endoplasmic reticulum, alpha-1 and alpha-2 helices. These form peptide chains have registration peptides on each end and a signal peptide. Polypeptide chains are released into the lumen of the RER. Signal peptides are cleaved inside the RER and the chains are now known as pro-alpha chains. Hydroxylation of lysine and proline amino acids occurs inside the lumen. This process is dependent on ascorbic acid as a cofactor. Glycosylation of specific hydroxylysine residues occurs. Triple E-bound helical structure is formed inside the endoplasmic reticulum from each two alpha-1 chains and one alpha-2 chain. Precollagen is shipped to the Golgi apparatus, where it is packaged and secreted by exocytosis. Outside the cell, registration peptides are cleaved and tropocollagen is formed by precollagen peptidase. Multiple tropocollagen molecules form collagen fibrils, via covalent cross-linking by lysyl oxidase which links hydroxylysine and lysine residues. Multiple collagen fibrils form into collagen fibers. Collagen may be attached to cell membranes via several types of protein, including fibronectin and integrin. Synthetic pathogenesis. Vitamin C deficiency causes scurvy, a serious and painful disease in which defective collagen prevents the formation of strong connective tissue. Gums deteriorate and bleed, with loss of teeth. Skin discolors, and wounds do not heal. Prior to the 18th century, this condition was notorious among long-duration military, particularly naval, expeditions during which participants were deprived of foods containing vitamin C. An autoimmune disease such as lupus erythematosus or rheumatoid arthritis may attack healthy collagen fibers. Many bacteria and viruses secrete virulence factors, such as the enzyme collagenase, which destroys collagen or interferes with its production. Molecular structure. The tropocollagen or collagen molecule is a subunit of larger collagen aggregates such as fibrils. At approximately 300 nm long and 1.5 nm in diameter, it is made up of 3 polypeptide strands, each possessing the conformation of a left-handed helix. These 3 left-handed helices are twisted together into a right-handed triple helix or superhelix, a cooperative quaternary structure stabilized by numerous hydrogen bonds. With type I collagen and possibly all fibrillar collagens if not all collagens, each triple helix associates into a right-handed super-coil referred to as the collagen microfibril. Each microfibril is interdigitated with its neighboring microfibrils to a degree that might suggest they are individually unstable, although within collagen fibrils, they are so well ordered as to be crystalline. A distinctive feature of collagen is the regular arrangement of amino acids in each of the 3 chains of these collagen subunits. The sequence often follows the pattern GlyPro-X or GlyX-Hip, where X may be any of various other amino acid residues. Proline or hydroxyproline constitute about 1/6 of the total sequence. With glycine accounting for the 1/3 of the sequence, this means approximately half of the collagen sequence is not glycine, proline or hydroxyproline, a fact often missed due to the distraction of the unusual GX1-X2 character of collagen alpha peptides. The high glycine content of collagen is important with respect to stabilization of the collagen helix as this allows the very close association of the collagen fibers within the molecule, facilitating hydrogen bonding and the formation of intermolecular cross-links. This kind of regular repetition and high glycine content is found in only a few other fibrous proteins, such as silk fibroin. About 75–80% of silk is gliala with 10% serine, and elastin is rich in glycine, proline, and alanine, whose side group is a small methyl group. Such high glycine and regular repetitions are never found in globular proteins save for very short sections of their sequence. Chemically reactive side groups are not needed in structural proteins, as they are in enzymes and transport proteins. However, Collagen is not quite just a structural protein. Due to its key role in the determination of cell phenotype, cell adhesion, tissue regulation and infrastructure, many sections of its non-proline-rich regions have cell or matrix association/regulation roles. The relatively high content of proline and hydroxyproline rings, with their geometrically constrained carboxyl and amino groups, along with the rich abundance of glycine, accounts for the tendency of the individual polypeptide strands to form left-handed helices spontaneously, without any intracane hydrogen bonding. Because glycine is the smallest amino acid with no side chain, it plays a unique role in fibrous structural proteins. In collagen, Gly is required at every third position because the assembly of the triple helix puts this residue at the interior of the helix, where there is no space for a larger side group than glycine unregistered trademark S single hydrogen atom. For the same reason, the rings of the pro and hip must point outward. These two amino acids help stabilize the triple helix hip even more so than pro. A lower concentration of them is required in animals such as fish, whose body temperatures are lower than most warm-blooded animals. Lower proline and hydroxyproline contents are characteristic of cold water, but not warm water fish. The latter tend to have similar proline and hydroxyproline contents to mammals. The lower proline and hydroxyproline contents of cold water fish and other poikilotherm animals leads to their collagen having a lower thermal stability than mammalian collagen. This lower thermal stability means that gelatin derived from fish collagen is not suitable for many food and industrial applications. The Tropocollagen subunits spontaneously self-assemble, with regularly staggered ends, into even larger arrays in the extracellular spaces of tissues. In the fibrillar collagens, the molecules are staggered from each other by about 67 nm. Each D period contains 4 plus a fraction collagen molecules, because 300 nm divided by 67 nm does not give an integer. Therefore, in each D period repeat of the microfibril, there is a part containing 5 molecules in cross-section, called the A-E overlap A-, and a part containing only 4 molecules, called the gap. The triple helices are also arranged in a hexagonal or quasi-hexagonal array in cross-section, in both the gap and overlap regions. There is some covalent cross-linking within the triple helices, and a variable amount of covalent cross-linking between tropocollagen helices forming well-organized aggregates. Larger fibrillar bundles are formed with the aid of several different classes of proteins, glycoproteins and proteoglycans to form the different types of mature tissues from alternate combinations of the same key players. Collagen's insolubility was a barrier to the study of monomeric collagen until it was found that tropocollagen from young animals can be extracted because it is not yet fully cross-linked. However, advances in microscopy techniques and atomic force microscopy, and X-ray diffraction have enabled researchers to obtain increasingly detailed images of collagen structure in situ. These later advances are particularly important to better understanding the way in which collagen structure affects cell-cell and cell-matrix communication, and how tissues are constructed in growth and repair, and changed in development and disease. For example, using AFM-based nanoindentation, it has been shown that a single collagen fibril is a heterogeneous material along its axial direction with significantly different mechanical properties in its gap and overlap regions, correlating with its different molecular organizations in these two regions. Collagen fibrils aggregates are arranged in different combinations and concentrations in various tissues to provide varying tissue properties. In bone, entire collagen triple helices lie in a parallel, staggered array. 40 nm gaps between the ends of the tropocollagen subunits probably serve as nucleation sites for the deposition of long, hard, fine crystals of the mineral component, which is Ca10, OH, 2, PO4, 6. Type I collagen gives bone its tensile strength. Types and associated disorders. Collagen occurs in many places throughout the body. Over 90% of the collagen in the body, however, is type I. So far, 28 types of collagen have been identified and described. The 5 most common types are collagen I, skin, tendon, vascular ligature, organs, bone; collagen II, cartilage; Collagen III, reticulate, commonly found alongside type I. Collagen IV, forms basal lamina, the epithelium-secreted layer of the basement membrane. Collagen V, cell surfaces, hair and placenta. Collagen-related diseases most commonly arise from genetic defects or nutritional deficiencies that affect the biosynthesis, assembly, post-translational modification, secretion, or other processes involved in normal collagen production. In addition to the above-mentioned disorders, excessive deposition of collagen occurs in scleroderma. Diseases: 1,000 mutations have been identified in 12 out of more than 20 types of collagen. These mutations can lead to various diseases at the tissue level. Osteogenesis imperfecta caused by a mutation in type I collagen, dominant autosomal disorder, results in weak bones and irregular connective tissue. Some cases can be mild while others can be lethal. Mild cases have lowered levels of collagen type I while severe cases have structural defects in collagen. Kandra Dysplasia is a uroskeletal disorder believed to be caused by a mutation in type II collagen. Further research is being conducted to confirm this. Ehlers-Danlos syndrome. 10 different types of this disorder, which lead to deformities in connective tissue. Some types can be lethal, leading to the rupture of arteries. Each syndrome is caused by a different mutation. For example, type 4 of this disorder is caused by a mutation in collagen type 3. Alport Syndrome. Can be passed on genetically, usually as X-linked dominant, but also as both an autosomal dominant and autosomal recessive disorder. Sufferers have problems with their kidneys and eyes. Loss of hearing can also develop during the childhood or adolescent years. Osteoporosis. Not inherited genetically, brought on with age, associated with reduced levels of collagen in the skin and bones. Growth hormone injections are being researched as a possible treatment to counteract any loss of collagen. Knobloch Syndrome. Caused by a mutation in the collagen 18 gene, patients present with protrusion of the brain tissue and degeneration of the retina. An individual who has family members suffering from the disorder are at an increased risk of developing it themselves as there is a hereditary link. Characteristics: collagen is one of the long, fibrous structural proteins whose functions are quite different from those of globular proteins, such as enzymes. Tough bundles of collagen called collagen fibers are a major component of the extracellular matrix that supports most tissues and gives cell structure from the outside, but collagen is also found inside certain cells. Collagen has great tensile strength, and is the main component of fascia, cartilage, ligaments, tendons, bone and skin. Along with soft keratin, it is responsible for skin strength and elasticity, and its degradation leads to wrinkles that accompany aging. It strengthens blood vessels and plays a role in tissue development. It is present in the cornea and lens of the eye in crystalline form. Uses: collagen has a wide variety of applications, from food to medical. For instance, it is used in cosmetic surgery and burn surgery. It is widely used in the form of collagen casings for sausages, which are also used in the manufacture of musical strings. If collagen is subject to sufficient denaturation, for example by heating, the three tropocollagen strands separate partially or completely into globular domains, containing a different secondary structure to the normal collagen polyproline II, for example random coils. This process describes the formation of gelatin, which is used in many foods, including flavored gelatin desserts. Besides food, gelatin has been used in pharmaceutical, cosmetic, and photography industries. From a nutritional point of view, collagen and gelatin are a poor-quality sole source of proteins since they do not contain all the essential amino acids in the proportions that the human body requires. They are not complete proteins. Manufacturers of collagen-based dietary supplements claim that their products can improve skin and fingernail quality as well as joint health. However, mainstream scientific research has not shown strong evidence to support these claims. Individuals with problems in these areas are more likely to be suffering from some other underlying condition rather than just a protein deficiency. From the Greek for glue, kolla, the word collagen means glue producer, and refers to the early process of boiling the skin and sinews of horses and other animals to obtain glue. Collagen adhesive was used by Egyptians about 4,000 years ago, and Native Americans used it in bows about 1,500 years ago. The oldest glue in the world, carbon dated as more than 8,000 years old, was found to be collagen used as a protective lining on rope baskets and embroidered fabrics, and to hold utensils together. Also in crisscross decorations on human skulls. Collagen normally converts to gelatin, but survived due to dry conditions. Animal glues are thermoplastic, softening again upon reheating, and so they're still used in making musical instruments such as fine violins and guitars, which may have to be reopened for repairs. An application incompatible with tough, synthetic plastic adhesives, which are permanent. Animal sinews and skins, including leather, have been used to make useful articles for millennia. Gelatin resocinyl formaldehyde glue has been used to repair experimental incisions in rabbit lungs. Medical uses, cardiac applications, the four collagenous valve rings, the central body of the heart and the extended cardiac skeleton of the heart are histologically and uniquely bound to cardiac muscle. Collagen is the floor of the atria joining the ceiling of the ventricles. Collagen contribution to cardiac performance summarily represents an essential, unique and moving solid form of mass opposed to the fluid mechanics of blood mass movement within the heart. The collagenous structure that divides the upper chambers of the heart from the lower chambers is an impermeable firewall that excludes both blood and electrical influence through customary anatomical channels. Thanks to collagen, atrial fibrillation almost never deteriorates to ventricular fibrillation. Collagen is infiltrated in varying densities with cardiac muscle mass. The amount, distribution, age and density of collagen all contribute to the compliance required to move blood back and forth. Individual cardiac valvular leaflets are forged into shape by specialized collagen under variable pressure. Gradual calcium deposition within collagen occurs as a natural consequence of aging. Calcium-rich fixed points within collagen and a moving display of blood and muscle enables methods of cardiac imaging technology to arrive at ratios essentially stating blood in and blood out. Pathology of the collagen underpinning of the heart is closely related to the category connective tissue disease. Type 2 collagen and rheumatoid arthritis, according to a study published in the journal Science, oral administration of type 2 collagen improves symptoms of rheumatoid arthritis. The authors conducted a randomized, double-blind trial involving 60 patients with severe, active rheumatoid arthritis. A decrease in the number of swollen joints and tender joints occurred in subjects fed with chicken type 2 collagen for 3 months, but not in those that received a placebo. 4 patients in the collagen group had complete remission of the disease. No side effects were evident. Hydrolyzed type 2 collagen and osteoarthritis. A published study reports that ingestion of a novel low molecular weight hydrolyzed chicken sternal cartilage extract, containing a matrix of hydrolyzed type 2 collagen, chondroitin sulfate, and hyaluronic acid, marketed under the brand name BioCell Collagen, relieves joint discomfort associated with osteoarthritis. A randomized controlled trial enrolling 80 subjects demonstrated that BioCell Collagen was well tolerated with no serious adverse event and led to a significant improvement in joint mobility compared to the placebo group on days 35 and 70. Cosmetic surgery. Collagen has been widely used in cosmetic surgery, as a healing aid for burn patients for reconstruction of bone and a wide variety of dental, orthopedic, and surgical purposes. Both human and bovine collagen is widely used as dermal fillers for treatment of wrinkles and skin aging. Some points of interest are, when used cosmetically, there is a chance of allergic reactions causing prolonged redness. However, this can be virtually eliminated by simple and inconspicuous patch testing prior to cosmetic use, and, most medical collagen is derived from young beef cattle from certified BSE-free animals. Most manufacturers use donor animals from either closed herds, or from countries which have never had a reported case of BSE such as Australia, Brazil, and New Zealand. Porcine tissue is also widely used for producing collagen sheet for a variety of surgical purposes, alternatives using the patient's own fat, hyaluronic acid, or polyacrylamide gels which are readily available. Bone grafts. As the skeleton forms the structure of the body, it is vital that it maintains its strength, even after breaks and injuries. Collagen is used in bone grafting as it has a triple helical structure, making it a very strong molecule. It is ideal for use in bones, as it does not compromise the structural integrity of the skeleton. The triple helical structure of collagen prevents it from being broken down by enzymes, it enables adhesiveness of cells, and it is important for the proper assembly of the extracellular matrix. Tissue regeneration. Collagen scaffolds are used in tissue regeneration either in sponges, thin sheets or gels. Collagen has the correct properties for tissue regeneration such as pore structure, permeability, hydrophilicity, and it is stable in vivo. Collagen scaffolds are also ideal for the deposition of cells, such as osteoblasts and fibroblasts, and once inserted, growth is able to continue as normal in the tissue. Reconstructive surgical uses. Collagens are widely employed in the construction of the artificial skin substitutes used in the management of severe burns. These collagens may be derived from bovine, equine, porcine, or even human sources, and are sometimes used in combination with silicons, glycosaminoglycans, fibroblasts, growth factors, and other substances. Collagen is also sold commercially in pill form as a supplement to aid joint mobility. However, because proteins are broken down into amino acids before absorption, there is no reason for orally ingested collagen to affect connective tissue in the body, except through the effect of individual amino acid supplementation. Collagen is also frequently used in scientific research applications for cell culture, studying cell behavior and cellular interactions with the extracellular environment. Wound care management uses. Collagen is one of the body unregistered trademark key natural resources and a component of skin tissue that can benefit all stages of the wound healing process. When collagen is made available to the wound bed, closure can occur. Wound deterioration, followed sometimes by procedures such as amputation, can thus be avoided. Collagen is a natural product, therefore it is used as a natural wound dressing and has properties that artificial wound dressings do not have. It is resistant against bacteria, which is of vital importance in a wound dressing. It helps to keep the wound sterile, because of its natural ability to fight infection. When collagen is used as a burn dressing, healthy granulation tissue is able to form very quickly over the burn, helping it to heal rapidly. Throughout the 4 phases of wound healing, collagen performs the following functions in wound healing: guiding function. Collagen fibers serve to guide fibroblasts. Fibroblasts migrate along a connective tissue matrix. Chemotactic properties. The large surface area available on collagen fibers can attract fibrogenic cells which help in healing. Nucleation. Collagen, in the presence of certain neutral salt molecules, can act as a nucleating agent causing formation of fibrillar structures. A collagen wound dressing might serve as a guide for orienting new collagen deposition and capillary growth. Hemostatic properties. Blood platelets interact with the collagen to make a hemostatic plug. See also, hydrolyzed collagen, a common form in which collagen is sold as a supplement. Animal glue, gelatin, fibrous protein, osteoid, collagen-containing component of bone, lysyl oxidase and LOXL1, LOXL2, LOXL3, LOXL4 in collagen formation, collagenase, the enzyme involved in collagen breakdown and remodeling. For more on other proteases that target collagen, see the proteolysis map. Ehlers-Danlos syndrome, hypermobility syndrome, sponge, cell types, drug discovery and development of MMP inhibitors, references. External links: 12 types of collagen, database of type I and type III collagen mutations, Science Durbix collagen, collagen stability calculator, computer-generated animations of the assembly of type I and type IV collagens, integrin collagen interface, PMAPA Euro animation, integrin collagen binding model, PMAPA Euro animation, collagen integrin atomic detail, PMAPA Euro animation.